continue with collagen. Collagen is actually a most abundant protein in the human body and it is synthesized by connective tissue cells. So this collagen is of four types. Type 1, in the 1 you can see the bone spelling, you can see 1. In tendon you can see 1. So bone and tendon have type 1 collagen. Type 2 collagen is in cart 2, you can see cartilage, right? 2. So cartilage. Type 3 is actually in the blood vessels. So in Ehlers-Danlos syndrome, especially of vascular type, you have deficiency of type 3 collagen. And type 4 collagen is under the floor, that is nothing but in the basement membrane. So actually in Alport syndrome, which is hereditary nephritis, here you find deficiency of type 4 collagen. And one more thing, these three types, type 1, type 2, type 3 collagens are fibrillar, that is they are having a capacity to assemble into fibrils. Now coming to collagen synthesis and structure. Actually collagen is nothing but a protein, so it is synthesized in our rough endoplasmic reticulum. So the first process is synthesis. Actually the collagen is pre-pro-collagen. So this pre-pro-collagen should be converted to pro-collagen inside the cell. So what's the process? Let's see. The first process is synthesis. Actually collagen is made up of 3 alpha chains. So pro alpha chain backbone, the first thing we need to form for the formation of pro collagen is we need to have the backbone, that is the alpha chain, and it contains glyxy. Here glyxy represents gly for glycine, X is proline and lysine, and Y is hydroxylated proline and hydroxylated lysine. So this is the first synthesis process. After synthesis we need to add hydroxyl ends. You can see the hydroxyl, that is process called as hydroxylation. So we need to hydroxylate proline and lysine, and next after hydroxylation there's a process of glycosylation. In the name itself you can see glycosylation, nothing but adding sugar molecules. So we start to add sugars in this third process, and adding of this sugars via hydrogen and disulfide bonds. We need to form a hydrogen and disulfide bond to form a procollagen, which is a triple helix molecule. So after glycosylation we need to form a hydrogen and disulfide bond via procollagen, so that we can form a triple helix of alpha chains, which is a procollagen. So one thing important in hydroxylation is this process requires vitamin C. So if there is no vitamin C, like in cases like scurvy, then this process may stop. So vitamin C is important here, and here alpha chain we need glycine, lysine, proline, okay. And here in glycosylation we add sugar. After that by formation of hydrogen disulfide bond we form the procollagen, which is a triple helix. Now after procollagen forming here in the rough endoplasmic reticulum, it is transported to Golgi, and from the Golgi it will undergo exocytosis process and finally comes out of the cell. So after coming out of the cell there is a process called proteolytic process. Proteolytic means we cleavage the disulfide ends of procollagen and form an insoluble tropocollagen. So procollagen is soluble, so that's why it is soluble through this membrane and came out. But now we are forming insoluble tropocollagen, so that again it will not go back into the cell, okay. So in the extracellular space tropocollagen we remove the C and N terminals. After that there is a process called assembly and alignment. You can see the assembly, that is these collagen molecules they assemble in fibrils and they are ready for cross-linking. So the last step is cross-linking, and this cross-linking is stabilized by enzyme called lysyl oxidase, and this lysyl oxidase also require copper for this process of cross-linking, and after cross-linking finally collagen fiber is formed. And this actually there's a problem in cross-linking in the case of Menkes disease. In Menkes disease you don't have this cross-link process, so that the collagen fiber don't form there, and that will lead to some problems. We'll discuss that in next page. Now let's continue with different diseases. Scleroderma is a disease where there is overproduction of collagen, so it is leading to hardening of tissue. It is actually autoimmune disorder. Next, coming to aging. In aging, there are more wrinkles because of decreased elastin and also decreased collagen production in dermis. So collagen is important to make a skin tight. If it is more, of course, it will harden the tissue in the case of scleroderma. Now coming to the disease called osteogenesis imperfecta. So in the name itself you can see osteogenesis. So here you can guess collagen type 1 is defective. As collagen type 1, that is bone, there is bone is type 1 collagen. So type 1 collagen is defective here. And one more thing, what cycle of collagen is defective here is glycosylation process. If you remember in this diagram, glycosylation process is where you need to form the disulfide bridges and hydrogen to form a triple helix procollagen. So the problem in osteogenesis imperfecta is you're not able to produce a triple helix procollagen. So you're not forming this, especially type 1 collagen. So this is the problem here. And the genes which are causing this defect, there's a defect in a gene like COL1A1, COL1A2. So COL1A1, COL1A2 is a defect which is leading to this osteogenesis imperfecta. The problems are there are multiple fractures and bone deformities just after a minimal trauma. You can see during birth in children. And next one is blue sclera. You can see the blue sclera. It is due to translucent connective tissue over choroidal veins like this. And also it forms some tooth abnormalities like obsolete teeth that can wear easily and due to lack of dentin, that is dentinogenesis imperfecta. So actually dentin is also a bone, right? Even that is not formed. So teeth problems are there. In sclera you see blue sclera, and you also see hearing loss because of loss of ossicles. Ossicles are not present, so hearing loss. So the mnemonic is bite, that is bone problem, eye problem, teeth problem, and ear problem. And one more thing, type 2 collagen problems will lead to lethal problems in utero, whereas type 3 and type 4 are more severe than type 4, that is blood vessels vascular and also basement membrane problems are more severe than type 1, that is bone problems. So severity, if you see for collagen diseases, type 2 is more severe, that is cartilage problems are more severe, next coming to blood vessels, next coming to basement membrane, and finally bone. So now let us discuss about Ehlers-Danlos syndrome. You can see these diagrams. This is hyperextensible joint and hyperextensible skin. So here the problem is abnormal collagen synthesis. So that is leading to hyperextensible skin, hypermobile joints. So the skin here is called as velvety skin, and the scars may be formed called cigarette paper scars. And this Ehlers-Danlos syndrome may also cause mitral valve prolapse. Actually, this disease may be either autosomal dominant or recessive, but mostly this osteogenesis imperfecta is autosomal dominant. Okay, this may differ either autosomal dominant or recessive, and this is mainly associated with joint dislocations because of this mobility, berry aortic aneurysms, and also some organ rupture. Hypermobility type of joint instability is most common here in Ehlers-Danlos syndrome. And this syndrome is divided into two types. It can either be joint and skin type or vascular type. If you remember the vascular type, in the vascular type, type 3 collagen is absent, and in the classic type, there's mutation in type 5 collagen. So type 5 collagen, especially mutation in genes like COL5A1, COL5A2. Here in this genes call 1 represents type 1 collagen, and here in this disease Ehlers-Danlos you can see call 5 represents type 5 collagen problem in classical joint and skin type, whereas in vascular type you can understand call 3A1, 3 represents type 3 collagen, that is blood vessels are defective here. And remember that this vascular type of Ehlers-Danlos is most threatening form because of its involving aorta, muscles, organs which are prone to rupture like gravid uterus. So this is very important. So two types of Ehlers-Danlos syndrome. Remember this genes number is according to the type of collagen. So in osteogenesis imperfecta, call 1A1, 1A2. Here call 5A1, 5A2 classical type. Vascular type call 3A1. Now Menkes disease. Menkes disease is a problem already I said in the cross-linking of collagen, so that there's a defective especially enzyme like lysyl oxidase enzyme, because here the copper absorption is limited in Menkes disease. You know that copper is required for cross-linking along with lysyl oxidase. So here copper is not there. can see there's a mutation in ATP7A, that is absent copper in Menkes disease. That is the problem. Actually, it's a X-linked recessive disorder, connective tissue disease. If you compare this with another copper disease, that is Wilson's disease, where there is ATP7B, there is copper buildup in Wilson disease. Here there is copper deficiency. So there's no cross-linking in this disease, that is leading to kinky hair and growth and developmental delay, also hypotonia, more risk of cerebral aneurysm. So this kind of disease is fatal in childhood. Now let's continue with elastin. So elastin is a stretchy protein we know which is present in our skin, lungs, arteries, elastic ligaments, vocal cords, ligamenta flava. So all this are having elastin. So this elastin is actually rich in non-hydroxylated proline glycine lysine. So if you remember, collagen is also made up of proline glycine lysine, but they are hydroxylated there because of hydroxylation process in the presence of vitamin C, right? But here there is no hydroxylation process, so they are non-hydroxylated, so they are like elastic material. Okay, and the cross-linking here occurs extracellularly, same like collagen, by lysyl oxidase, and this gives a elastic property. So actually this elastic property can be broken down sometimes by it can be broken down by alpha 1 antitrypsin. But there's a disorder where there's an inherited disorder of not having alpha 1 antitrypsin. So in that case you can understand the elasticity may increase, like a rubber band, the elasticity may increase without inhibiting it if there is no alpha 1 antitrypsin. So that will cause a disease like COPD. Now let us discuss about Marfan syndrome. So in Marfan syndrome you need to remember the letter F more, because this is caused because of a FBN1 gene mutation on chromosome number 15F again, and here there is a defective fibrillin 1 protein. So F is most important, and this is actually a autosomal dominant disease. Okay, and it may involve skeleton, heart, eyes. So the problem is here the patient have tall, long extremities. This important my finding for Marfan syndrome is chest wall deformity called as pectus craniatum, that is a pigeon chest, also called as pectus excavatum. And this patient presents with hypermobile joints, long tapering fingers and toes called arcanodactyl, and cystic medial necrosis of aorta, aortic root aneurysm, rupture, dissection is the most common cause of death. So aortic root aneurysm, rupture and dissection, most common cause of death in Marfan syndrome. Mitral valve prolapse is also present. This increased risk of spontaneous pneumothorax and early age cataracts are also present in Marfan syndrome. The problem is defect in FBN1 gene in chromosome 15, so that is leading to defective fibrillin 1. Next, coming to homocystinuria. Actually, these symptoms are like similar to Marfan syndrome, but here the problem is due to cystothionein synthase deficiency. So this is leading to homocysteine buildup, and here the presentations are like Marfan syndrome, like pectus deformity, tall stature, arcanodactyl, joint hyperlaxicity. Then how to differentiate both from other? You can see here the differentiation. Marfan syndrome is dominant, autosomal dominant, homocystinuria is recessive. Intellectuality is normal because it's dominant. I remember the normal intellectual, but homocystinuria here recessive, so I think decrease intellectual ability. The vascular complications, aortic root dilation, here thrombosis. In lens discoloration, Marfan fans out. I said remember F for Marfan, FPN1 gene, 15 chromosome, fibrillin 1, and fans out, upward or temporal. But homocystinuria downward or nasal. So Marfanoid habitus means tall with long wingspan, long fingers, also seen in homocystinuria, meant to be syndrome, and Ehlers-Danlos syndrome. Next, coming to Williams syndrome. Actually here there's a partial deletion on long arm of chromosome 7. So you can see FN appearance and intellectual disability, and also supravalvular aortic stenosis. So this is about Williams syndrome. So we are done with molecular part of biochemistry, cellular part of biochemistry. Now we'll continue with the laboratory techniques of biochemistry. Laboratory techniques will begin with polymerase chain reaction. This process is required for finding the viral load in cases like HIV, herpes encephalitis, etc. So here we have three processes. The first one is denaturation. It is a heat sample. We heat the DNA, double-stranded DNA, and we break down them into single-stranded DNA. The second process is annealing. In annealing, we add a primer to the DNA so the replication process can begin. And the third one is elongation. In elongation is nothing but you warm the sample so that the DNA polymerase gets activated and start replicating, forming a new strand. So this is polymerase chain reaction. Here we need to know the temperatures. For denaturation, we heat the sample. For annealing, we cool the sample. And for elongation, we warm the sample. So heating and cooling cycles continue until the amount of DNA we want is sufficient. So we can also do real-time polymerase chain reaction in the presence of fluorescence dye. So that here in the live we can see if fluorescence increase, that indicates more DNA is formed in the sample. Now coming to CRISPR or Cas9. Actually this process is derived from the bacteria. We have a guide RNA here. You can see this is a guide RNA, and this is complementary to the DNA sequence which we form here. And also we have an endonuclease called Cas9. You can see the endonuclease Cas9, which makes single or double strand breaks at the target site. So this endonuclease is what which breaks this DNA. Okay. And what we do is, now we are dividing them into two types. One is non-homologous repair, one is homologous repair. Non-homologous repair we do for the one which are broke in an incorrect manner, or imperfectly cut segments are sent to non-homologous site. So if there are some accidental frame shift mutations, then just will remove this kind of protein. And next one is we do a donor DNA sequence. You can add a donor DNA sequence in homologous repair system. So here we'll add our desired DNA of homologous site so that we can form a edited sequence. So we will knock in, that means we accept this kind of repair here in this process. So this process actually we use it in removing some virulence factors from pathogens. We replace disease causing alleles of genes with healthy variants. So we use it in clinical trials of sickle cell disease, and also specifically we target tumor cells so that we can get the desired one. Now coming to blotting procedures, they are divided into southern, northern, western, southwestern. So southern block is the one which identifies the DNA. Actually what we do is we take a gel electrophoresis, we arrange the DNA, we cut them, we arrange them according to their size. And next what they do is their membrane will be exposed to the DNA probe, so that the DNA probe anneals according to its complementary strand. So it will you can see this red one, they are selected like that. The probe will identify the DNA according to its complementary strand. So finally we get a labeled piece of DNA, and so this is how we find the DNA by using probe. This is southern blot. We actually do this on multiple samples. Clinical uses like in sickle cell anemia, we want to find out the sickle cell anemia DNA, then we use that kind of probe. Let's come into northern blot. It is same like southern blot. Here also we use gel electrophoresis, same technique. But here in northern blot, we identify the RNA. Next in western blot, we identify the antibodies. So actually here we put a sample protein on gel electrophoresis, and now we label an antibody, and we'll see which antibody is binding to that protein so that we can find out the antibodies here. Or you can say you can find out the relevant protein here. So and one more thing, northern blots can also detect the splicing errors. Splicing process where we convert transcription to translation. In that process, splicing errors can be found by northern blot because they will identify RNA. Next coming to southwestern blot. In the name itself you can see you use south and western blot. So that is we can identify DNA and also protein. So here we identify the DNA binding protein like C-Jun, C-Phos using labeled double-stranded DNA probes. Now let's discuss about flow cytometry. In the name itself you can see flow cytometry means we allow the flow of cells and we allow that cell to be attached to that specific antibody for which it can be attached, and to that cell only we attach the fluorescent label and now allow them to flow. So now we put a laser light so we can see whichever antibody along with its protein which is labeled with fluorescent will shine. It will fluorescent, laser makes the label fluorescent, so that this can be detected by a detector. So it will count the number of I can see fluorescence labeled attached cells, and now this which are counted can be labeled by a histogram, or you can use a scatter plot. You can see here we are doing the scatter plot for CD8 positive and CD3 positive, of course also negative. So here you can see the cells in the left lower quadrant are negative for both CD8, they are negative for CD8 and also negative for CD3, whereas cells in the right lower quadrant, you can see actually there are no cells but it is positive for CD8 and negative for CD3. And next cells in the left upper quadrant, this one positive for CD3 and negative for CD8. Cells in the right upper quadrant are positive for both CD8 and CD3. So like this we can find out the different cells, and this is used in mainly fetal maternal hemorrhage to find out some monoclonal antibodies against to hemoglobin F, that is a fetal hemoglobin. So actually if the particles are moving forward, that means we can find we can assess the size of the particle. If the particles are scattering towards the side, we can assess the granularity. So this is about flow cytometry. So next coming to microarrays. So microarrays are like it's like a comparison test between two cycles. For example, you can take thousands of DNA oligonucleotides. They are actually arranged in a grid or glass like chip. Then this DNA or RNA samples are compared and attached to different fluorophores, and they are also hybridized so that we can see the ratio of fluorescence signal at particular oligonucleotide, which reflects the amount of hybridizing nucleic acid in two samples. So we can see which is hybridizing more nucleic acid. So this is mainly used to compare the transcription of genes either in RNA or two DNA samples. So we can detect even single nucleotide polymorphisms and copy number variants for genotyping. So it is actually used in clinical genetic testing, forensic analysis, cancer mutation, genetic linkage analysis when DNA is used. So we can compare two types of DNA or DNA with RNA or two types of RNA. So here cDNA is tested using microarray, which determines the gene expression. Now let's continue with ELISA, that is enzyme linked immunosorbent assay. Actually you need to understand this heading, then only you can understand this. It's enzyme link. So actually you have an antigen, and for that antigen you add some antibody. Okay, for that antibody if you add the enzyme, then this is nothing but ELISA test. Actually we use this to find out specific antigen or antibody in a patient's blood sample. So it is divided into direct type, indirect type and sandwich type. So let us see three types. The first one is direct type, where you have an antigen and for that antigen antibody is attached and for that enzyme is attached. So this is direct ELISA. Indirect ELISA is you have an antigen, there's antibody, and again upon that antibody you have another antibody, for that enzyme is linked. Then this is called indirect, because directly enzyme linked antibody is not present for this antigen. Next, sandwich. In the name itself you can see sandwich, like in between two antibodies you have a antigen. So first antibody, next antigen, again antibody, and this is actually an indirect antibody because upon one antibody you are having another antibody which is linked to enzyme. So this is enzyme linked immunosorbent assay. And when important is sandwich immunosorbent assay as high specificity. And next coming to competitive ELISA test is where you have an antigen like this, and you also have an antibody and some free antibodies present. So now that free antibody if added to some other sample, it can easily bind to antigens. So if there are more antigens, then you will get less color because more antigens are bound to this antibody, so free antigens are not present, so less color. But if there are less antigens, because of this free antibody, less antigens means still more antibodies are remained, so that you can get more color. So this is about ELISA. Next, coming to karyotyping. So, karyotyping is done especially to find out autosomal trisomies, sex chromosome disorders, or some chromosomal imbalance. So, we use a material called colchicine, which will halt the chromosomes in metaphase of cell cycle, so that we can stain them, we can order, number them according to morphology, size, arm length, ratio, banding patterns, so that we can find out any abnormalities, because normal cells will also be there and abnormal, so we can compare and contrast and identify the abnormal cells. So this can be done on a sample of blood or bone marrow, even from amniotic fluid in a pregnant female, and also placental tissue. So next is fish technique, that is fluorescence in situ hybridization. So actually this procedure is also same like karyotyping, but here we see different uh, chromosomes by means of coloring mechanism, that is fluorescence. So here actually it's divided into microdeletion, translocation, duplication. Microdeletion means actually the small part of that chromosome is deleted. So if you want to see the fluorescence there, you are not able to see, that is microdeletion. Next, translocation, for example, you can see this is a 17 chromosome which is yellowish color, not 17, this is 17 which is pinkish color, but here some upper part is missing. It got translocated into 19 chromosome, which you can see here, double white arrow here upward. So this is translocation. So all this you can see, microdeletions you can find out, translocations in chromosome you can find out, and duplication you can see this duplicated. This orange are two, actually they become four, that is tetrasomy. This is because of duplication. So all these processes you can find out clearly because of these colors, nice colors, you can find out this by means of fish technique where you use fluorescence. Last part of laboratory technique is molecular cloning. Actually it's very interesting part, that is we make recombinant DNA in the bacteria. Actually those are produced from the human, but we insert them by means of plasmid to the bacteria and grow them, so it is useful for us to form many types of DNA. So actually the first step what you need to do is you need to pick up the mRNA, especially from the eukaryote. So after taking this mRNA, you need to add reverse transcriptase. So by adding reverse transcriptase, now this mRNA will easily form DNA, especially it will form C type of DNA, which doesn't have any introns, which is very good because the splicing process is also like almost done. So we get nice DNA without introns. So now what we do is we take this cDNA and we insert them into bacterial plasmid, which contains especially antibiotic resistant strain, because if we give some antibiotic again it may become uh, this bacteria may die. So we take especially antibiotic resistant gene and we give this cDNA. So now we transform this plasmid into bacteria. Finally this bacteria will survive it on some antibiotic medium, and so that we can produce a clone DNA, copies of cDNA nicely of human form from the bacteria. So this is a very nice technique, molecular cloning. So by this we are done with laboratory techniques. Next we'll move on with